Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 29. The topic is polypeptides. Discussing agenda for the topic is making a polypeptide chain, protein structure and function, lecture outcome. After watching and listening this lecture, students will know about the process which leads to the formation of polypeptide from amino acid monomers. So basically we will try to know how the amino acid monomers combine and produce a polypeptide chain, what type of chemical interaction is involved, what type of a bond is formed between which atoms. Introduction. Now that we have examined amino acids in our last lecture, let's see how they are linked to form a polymer. Polymer is a molecule made up of several identical repeating units or monomers. So on the right side of your screen you can see a peptide bond in formation. Peptide bond is formed between the amino group of one amino acid and the carboxyl group of another amino acid. The peptide bond is basically a covalent bond, and it is formed with the removal of a water molecule. So the reaction overall is a condensation reaction or a dehydration reaction in which the water molecule is omitted, and a peptide bond is established between two amino acids. Remember that in the formation of a peptide bond, the skeleton of the two amino acids is involved, and there is no interaction between their side chains. When two amino acids are positioned so that the carboxyl group of one is adjacent to the amino group of the other, they can become joined by a dehydration reaction with the removal of a water molecule. The resulting covalent bond is called a peptide bond. So a peptide bond links two amino acids. Repeated over and over, this process yields a polypeptide, a polymer of many amino acids linked by peptide bonds. The repeating sequence of atoms highlighted in purple in figure 1 is called the polypeptide backbone. So this is the polypeptide backbone. These are the side chains of those amino acids, and the bond is formed between the carboxyl group of one amino acid and the amino group of another amino acid. Water molecule is excluded and a peptide bond is established, which is a covalent bond. Extending from this backbone are the different side chains. These are the side chains, we have already talked about it, and they give characteristic properties to amino acids. Polypeptides range in length from a few amino acids to 1000 or more amino acids. So they can be large in size depending upon the number of amino acids involved. Each specific polypeptide has a unique linear sequence of amino acids. Now that makes the proteins different from one another. The proteins are made up of 20 different types of amino acids, but there is enormous diversity in proteins. So this diversity is attributed to the fact that the sequence of the amino acids in different proteins is different, and secondly, the three-dimensional structure in which the proteins mold themselves is also unique to proteins. So these are two different properties which makes proteins distinct from one another: the sequence of the amino acids and the three-dimensional structure in which they are molded. Now, a polypeptide is a sequence or linear sequence of amino acids. A polypeptide has two distinct ends. On one end, there will be a free carboxyl group, and that end will be called C terminus because of the presence of the carboxyl group, and the other end will be having a free amino group, and because of that it is termed as N terminus. So a polypeptide has two distinct ends, one with a free carboxyl group called C terminus, the other with a free amino group called N terminus. The chemical nature of molecule as a whole is determined by the kind and sequence of the side chains. We have already discussed in our previous lecture that the side chain or the R group of each amino acid is unique, and it gives characteristic properties to that amino acid. Now when you discuss proteins, so the R groups also participate there, and the composition of these amino acids and their R groups give characteristic properties to that protein. The R groups determine how a polypeptide folds and thus its final shape and chemical characteristics. In the tertiary structure of proteins, we will discuss different types of interactions where the R groups are involved. There is covalent interactions, ionic interactions, disulfide bridges, van der Waals forces, hydrophobic interactions. So all those interactions takes place in the tertiary structure of proteins, and the side chains of the amino acids are involved in those interactions. So these side chains participate and they determine the overall shape of a protein. The immense variety of polypeptides in nature illustrate an important concept introduced earlier. that cells can make many different polymers by linking a limited set of monomers into diverse sequences. Recall from the previous slide where we said that the linear sequence of amino acids is different in different proteins. So this is an ability of the cell to form different types of proteins from a small number of building blocks by changing the sequences. Obviously, the sequences in the genes are different, and because the genes have message for the synthesis of a polypeptide chain, so the sequence of nucleotides in DNA determines the sequence of amino acids in proteins. The specific activities of proteins result from their intricate three-dimensional architecture, the simplest level of which is the sequence of their amino acids. Now, proteins have four structural levels, which we will discuss in the subsequent slides. But here you need to remember one thing, that the sequence of amino acids and the type of R groups determines which type of interaction will take place between the amino acids when this protein is molded into its secondary structure or tertiary structure or quaternary structure. So basically the side chains determine the type of shape the protein will finally adopt. What can the amino acid sequence of a polypeptide tell us about the three-dimensional structure, commonly referred to simply as the structure of the protein and its function? The term polypeptide is not synonymous with the term protein. We have already discussed that polypeptide is usually referred to a linear sequence of amino acids or a single chain, whereas a protein can be made up of more than one polypeptide, like in hemoglobin, which is made up of four different polypeptides. Even for a protein consisting of a single polypeptide, the relationship is somewhat analogous to that between a long strand of yarn and a sweater of particular size and shape that can be knitted from the yarn. So here they have given an example from the daily life where a single strand of yarn and a sweater which consists of thousands of strands. So you can refer to a single strand of yarn as a polypeptide and the sweater as a protein, where a protein is made up of several polypeptide chains. A functional protein is not just a polypeptide chain, but one or more polypeptides, precisely twisted, folded, and coiled into a molecule of unique shape, which can be shown in several different types of models. So a functional protein is composed of different types of polypeptides, where these polypeptides are twisted, folded, coiled in different unique ways. So in our next slide, we will study different types of models of proteins, which will give you an idea about their structural diversity. Here are three different types of models. Obviously, these models are computer generated models, and here they have shown lysozyme protein. These are three different models of a lysozyme protein. It is a protein present in the tears and saliva, and its role is to prevent infection. Its mode of action is binding to target molecules on bacteria. Once they bind to bacteria with the help of the target molecules on bacteria, then they degrade the bacterial cells. So now we analyze those three different types of models of the lysozyme protein. The first is the space filling model. In this model, all the atoms are shown, all the atoms present in the protein are shown, so it gives this type of a structure. Here in the ribbon model, the atoms participating in the formation of the protein are not shown, but this structure refers to the folds and coils the protein undergo to form a three-dimensional structure. And this is another structural depiction of a protein, where you can see those thin threads. These are the side chains of the protein, and here is the target molecule, the bacterial cell, and the virus. The lysosome has a specific binding site for this bacterial cell, and with the help of target molecules on the bacterial cell, the interaction or the complementarity is established, and thereby the bacterial cell is broken down. And it is the amino acid sequence of each polypeptide that determines what three-dimensional structure the protein will have under normal cellular conditions. In primary structure, there is peptide bond. Primary structure is a linear sequence of amino acids. And then in secondary structure, we have alpha helix and beta pleated sheets, where the skeleton of the amino acids is involved, and the R group does not participate. And then in the tertiary structure, the R group participate in forming a variety of interactions with the R groups of the other amino acids in the chain. And then finally in the quaternary structure, we have two different or two same polypeptides interacting with one another. So which type of interaction will be established, what will be the final form of a protein, it is determined by the side chains or the R groups of the amino acids which form a polypeptide chain. When a cell synthesizes a polypeptide, the chain may fold spontaneously, assuming the functional structure for that protein. Remember, when we were studying the rough endoplasmic reticulum, we talked about the molecular chaperones, which are responsible for the folding of proteins into their correct three-dimensional form. And if a protein is not folded properly in its 3D structure, then such a protein is tagged with ubiquitin and is directed towards degradation pathway, where the proteasomes participate and carry out the process of degradation. This folding is driven and reinforced by the formation of various bonds between parts of the chain, which in turn depends on the sequence of amino acids. Once again, we are highlighting the importance of the side chains again and again, because they participate in the type of interactions which will take place in the tertiary structure. Many proteins are roughly spherical. They are called globular proteins, where other proteins are shaped like long fibers. They are called the fibrous proteins, like the actin and myosin. And here you can give the example of hemoglobin. Even within these broad categories, countless variation exist. Now here is the simplified diagrams which you quite often see in your biology books. These are also examples of the structural diversity the proteins exhibit, and in order to make things simple and understandable, so these structures are given in various books. This is the transparent shape of a protein, and it is made up of polypeptide chains. This is a rhodopsin protein, which is present in the eyes. This is another protein which is a integral protein in the plasma membrane. These are the enzymes, biological catalysts, and finally these are the pancreas cells secreting insulin, which is responsible for breakdown of glucose. A protein's specific structure determines how it works. Structure, the proper three-dimensional structure of a protein is the key to the function it performs. Why mutations are harmful? A gene having thousands of sequences, when undergoes a mutation at a single position, and even only a single amino acid is replaced with another amino acid having different types of chemical characteristics, then even this small change have profound effect on the overall shape of the protein, because the side chain of almost every amino acid have some role in the development of the three-dimensional structure of proteins. So here you have an example how much the structure of a protein is important to the function it performs. On one side you have an antibody. Antibodies are proteins which are produced by the immune system against the invading pathogens. On the other side you have a flu virus. This is a protein from a flu virus. And if the antibody produced by the immune cells is formed properly in its proper three-dimensional structure, only then it can bind with the protein produced by the flu virus or the flu virus and neutralize the virus. If the three-dimensional structure is not properly developed because of a mutation or other errors, then this complementarity, this interaction cannot be established, and therefore this antibody cannot perform the role assigned to it. So the structure is the key to the function a protein performs. In almost every case, the function of a protein depends on its ability to recognize and bind to some other molecules. I think you will understand this statement better when you recall the enzymes. The enzymes have specific sites called active sites for the substrate. When the active site is distorted, then this particular enzyme cannot bind correctly with the substrate and therefore cannot perform the function assigned to it. Now we have already discussed this figure. And yet another astonishing example is endorphin molecules. Endorphin molecules are produced by pituitary glands, and these endorphin molecules, they attach to the pain receptors in the brain, and as a result the person feels relaxed and the pain is relieved. And you may have heard that several sedative agents like morphine or opium, they also bring about this action. So how a synthetic or a natural agent synthesized outside the body induces an effect which is caused by molecules produced inside the body. How an exogenous molecule induces an effect in the body which is attributed to some endogenous molecules. It is because of the similarity in the shapes. So this is the shape of an endorphin molecule, and on the other hand there is a shape of the morphine molecule. So naturally endorphin molecules binds to the pain receptors and the pain is relieved and the person feels satisfaction. On the other hand, morphine, which is an exogenous molecule, it mimics, it looks like endorphin, and therefore it brings about the same effect which is attributed to endorphin. So this is yet another astonishing example of the fact that the three-dimensional structure of a protein is vitally important for the function it performs. So this model gives you an idea about the fact that the correct 3D structure of a protein is important for the function it performs. With that, we come to the end of this lecture. This is the reference book from where those slides were prepared. I have already shared those chapters with you people. You can ask questions related to this topic on KCMS in that particular class activity, or you can ask questions via WhatsApp groups. Thank you for having me. Allah Hafiz.